Howdy students, this is Miss M yet again, and today we are looking at how to solve a dihybrid Punnett square. So the first thing I want you to do is put your name at the top of the paper. Now let's read the problem. Tall is dominant to short. Green hair is dominant to yellow hair. If a male elf that is homozygous tall and yellow hair is crossed with a heterozygous tall green-haired elf, what are the possible outcomes for the genotype and phenotype of the offspring? Okay, so the first thing we're going to do is determine the genotypes of the parents. For the female, it said that she is heterozygous tall, and we're going to assume this means that she's heterozygous for tall and green. So we're going to give her genotype as heterozygous tall and heterozygous green. For the male, we learned that he is homozygous tall and he has yellow hair. So we're going to make him homozygous for the dominant trait, which is tall, and we're going to give him yellow hair, and the only way he can have yellow hair is if he's homozygous for the recessive trait. We're going to create a key for these genotypes, so capital T represents tall, lowercase t represents short, capital G represents green, and lowercase g represents yellow. Now we're going to figure out the possible genotype combinations in the gametes of each parent, and we are not going to use the FOIL method. So we're going to mark this out. And the reason I don't like the FOIL method is because this problem is supposed to represent independent assortment of alleles. You see, these two alleles are on two different chromosomes that the female has. One was from her mother and the other one was from her father. Since they're two separate chromosomes, they can assort independently of each other. And so what we're going to do is we're going to consider that this allele from the female could sort with this allele from the male, or it could sort with this allele from the male. This chromosome from the female could assort with this allele from the male, or it could assort with this allele from the male. So when we're setting up our Punnett square, across the top here, we're going to put the different combinations that the tall trait can assort. So we could have capital T to capital T. We could have we could have capital T to capital T. Again. We could have lowercase t to capital T. Or we could have, again, lowercase t to capital T. And those are all of the different combinations for the height allele that we can have. For the color allele, same situation. From the mom, this allele could sort with this recessive, or it could sort with this recessive. So we could have dominant and recessive, or we could have dominant and recessive. This second allele from the mother could assort with this first color allele from the father, or it could sort with the second color allele from the father. So we could have homozygous recessive, or we could have homozygous recessive. Okay, now we're going to put the combinations together. So in the first box, we're going to have homozygous dominant for height. and heterozygous for color. And again, on the second box, homozygous dominant for height and heterozygous for color. Third box, homozygous dominant for height, but now we have homozygous recessive for color, so it would be yellow. In the fourth box here, again, homozygous dominant for height, and then homozygous recessive for color again. On the next row, the first combination, homozygous dominant to heterozygous, homozygous dominant to heterozygous, homozygous dominant to homozygous recessive, homozygous dominant to homozygous recessive. Now, when we're writing gene combinations, it is customary to put the capital first. The reason I put the lowercase first here is because I was showing that this allele would assort with either one of these alleles from the father. But when we're writing out the genotype, we need to put the capital first, the dominant trait first. So here we have heterozygous for height and heterozygous for color. Again, heterozygous for height, heterozygous for color, heterozygous for height, homozygous recessive for color, heterozygous for height, and homozygous recessive for color again. And our last set of combinations here are again heterozygous for height, heterozygous for color, heterozygous for height, heterozygous for color, heterozygous for height, homozygous recessive for color, heterozygous for height, and homozygous recessive for color. Okay, turn your paper over. And we're going to answer the questions on number 7, we're going to refer back to the box on the front side of the page. So, for the genotypes, if we look back at the boxes here, the first question is asking us, what's the percentage that are going to have homozygous for height and heterozygous for color? So if we look at the boxes here, we have 1, 2, 3, 4 out of the 16 boxes that are homozygous for height and heterozygous for color. So this first one is going to be 4 out of 16, which reduces to 1/4. So 1/4 of the genotypes are going to be capital T, capital T, capital G, lowercase g. The next combination we're asking about is homozygous for height and homozygous recessive for color. So let's look back at the box here. So we have 1, 2, 3, 4 that are homozygous dominant for height and homozygous recessive for color. So that's going to be 4 out of 16 again, which reduces to 1/4. So 1/4 of them are going to be homozygous dominant for height and homozygous recessive for color. Now we're looking for heterozygous for height and heterozygous for color. So let's go back to our box, our Punnett square, our dihybrid Punnett square. These are the combinations we're looking for. So we have 1, 2, 3, 4. So again, that's 4 out of 16, which reduces to 1/4. The last combination we're looking for the genotypes, those are the letters, remember, that represent the alleles, is heterozygous for height and homozygous recessive for color. And what do you think that is? 1, 2, 3, 4. So 4 out of 16 again, which is 1/4. Okay, so we can just double check here. We want 16 total, so 4 plus 4 plus 4 plus 4 is 16 out of 16, because we had 16 boxes in that dihybrid Punnett square. Okay, now we're looking at the phenotypes. So we're going to look at the Punnett square again, but now we're looking for tall and green. So to be tall, it has to have a capital T in it, and to be green, it has to have a capital G in it. So we're looking for homozygous dominant and heterozygous, or heterozygous and heterozygous. So let's go back to the Punnett square. We're looking for tall and green. So we have 1, 2, these are not tall and green, these are tall and yellow, 3, 4, 5, 6, 7, 8. So 8 out of the 16 are going to be tall and green. So that's 1/2 of them. A 1/2 possibility with each cross. Now we're looking for tall and yellow. So the only way we can be yellow is to be homozygous recessive. So we're looking for a capital T and 2 lowercase g's. So let's look back at our Punnett square. And capital T's and 2 lowercase g's. So here we have 1, 2, 3, 4, 5, 6, 7, 8. So that's going to be 8 out of 16 again, which is 1/2. So we have a 1/2 chance that the offspring is going to be tall, both tall and green, and a 1/2 chance that the offspring is going to be both tall and yellow. Well, what about short and green and short and yellow? Remember, to be short, it has to be homozygous recessive, 2 lowercase t's. Let's go back and look at that thing. Do we have 2 lowercase t's anywhere on that Punnett square? No, we do not. So there is a 0% chance that this offspring is going to be both short and green, or both short and yellow. Just not going to happen. All right, now let's look at another example. So in this case, we have a heterozygous in both coat color and coat texture male dog mating with a heterozygous color and texture female. Okay, how do I know that represents both color and texture? Well, capital B is black and lowercase b gives us brown. The recessive is brown. So it's heterozygous for color. Capital W, dominant, is going to be wiry hair. That's the texture of the hair. And lowercase w is smooth hair. So this dog, this male dog, is black with wiry hair. The female dog is also black with wiry hair. They're both heterozygous for both traits. Okay, so let's set this thing up again. We have heterozygous for color, heterozygous for texture, crossing with heterozygous for color and heterozygous for texture. And remember, these alleles from each parent can assort independently. So this B could assort with this B, or it could assort with this B. So the different combinations here are homozygous dominant, or heterozygous. This B could assort here, or it could assort here. So we have, again, heterozygous, and then homozygous recessive. For the texture of the hair, we could have this assortment or this assortment. So we have homozygous dominant or heterozygous, and we could have this assortment or this assortment. So we could have heterozygous or homozygous recessive. Okay, let's combine all of these alleles. Okay, these are supposed to be heterozygous. This one doesn't look that tall, but it's supposed to be. And then remember, it's customary to put the dominant first. So this is also a heterozygous combination. This one is homozygous recessive. All right. Got ahead of myself there. Okay, and then the last combinations. Okay, there we have our Punnett square. So now you may think we're done, but we're not. We are now going to figure out the genotypes and the phenotype possibilities from this cross. So let me move this over here, get the Punnett square to where we can see it. Okay, for the genotype possibilities. Now remember, the genotypes are these gene combinations. So we'll start with homozygous dominant for both color and texture. And if we look up at the Punnett square, we only have one of those. So that's going to be 1 out of 16. And for homozygous dominant and heterozygous for texture, we have 1, 2 of those. So that's going to be 2 out of a 16 possibility there. And for homozygous dominant and then homozygous recessive, we again have 1 out of 16 there. Okay, then for heterozygous for color and homozygous dominant for texture, we have 1, 2 of those, so there's a 2 in 16th possibility that the offspring will have that combination. For heterozygous for color and heterozygous for texture, we have 1, 2, 3, 4 boxes here, so we have 4 out of a 16 chance on that combination. And then for heterozygous for color but homozygous dominant for texture, we have 1, 2 out of 16 for that possibility. Let's reduce some of these fractions. This one is going to reduce down to 1/8 of a chance. This one is going to reduce down to 1/4. And 2/16 is again 1/8. And for our last set of combinations, homozygous recessive and homozygous dominant, for that combination we only have 1. So that's going to be a 1 in 16 chance that the offspring will look like that for homozygous recessive and then heterozygous on texture. We have 2 out of 16. Reduce that to 1/8 of a chance. And for our last gene combination for homozygous recessive on color, homozygous recessive on texture, we only have a 1 in 16 chance for that genotype. Okay, the last thing we're going to do on this sheet is look at the phenotypes, the possible phenotypes of this cross. All right, so the possible phenotypes are going to be a black dog with wire hair, or we could have a black dog with smooth hair, or we could have a brown dog with wire hair, or we could have a brown dog with smooth hair. Okay, so for black and wire hair, we need a capital B and a capital W. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9 out of 16 chance that it's going to be a black dog with wiry hair. Black dog with smooth hair, that's going to be a capital B and 2 lowercase w's. So we have 1, 2, 3 out of 16 chance that it's going to look like that. And then for a brown dog with wiry hair, we need 2 lowercase b's and a capital W. 1, 2, 3 out of 16 that it'll look like that. And then brown and smooth, we need 2 lowercase b's and 2 lowercase w's. And we just have 1 of those combinations. So 1 in 16 there. So let's just double check and make sure that we ended up with 16 total. There's 9, 10, 11, 12, 13, 14, 15, 16. So every possibility is accounted for here, and we can double check this one as well. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Every genotype possibility is accounted for there. Okay, so that's how you do a dihybrid cross. And you might see this one on the STAR test. This is something that we think they like to ask about. All right, we'll see you later.